Welcome and thank you for joining me today. My name is Katharina Nargar and I'm a botanist at the Australian Tropical Herbarium in National Research Collections, Australia. Today I want to provide you with a snapshot on where things are at in Australian plant taxonomy and provide you with some food for thought for our discussions on how we are going to describe the remaining Australian plants. The goals that we've set ourselves in the decadal plan for taxonomy and biosystematics in Australia and New Zealand are aspirational. And discovering and describing the remainder of Australian species within a generation, admittedly, is an incredibly ambitious aim, and also somewhat daunting. However, humankind has embarked on other great discovery missions before, so let's take a moment to consider the challenge of space exploration. Space missions excite and inspire the imagination of societies and governments. And looking at the bare surface of the moon, it may seem a little odd. However, a great part of the attraction is the fact that space missions always border at the impossible. Their pursuit requires a lot of creativity, high-tech and innovation, and the ability to think outside the box. And these expensive high-tech missions always hold the reward of demonstrating to the world what is possible, against all odds. Another reason for the great success and continued funding of these ambitious missions is that space programs deliver impact to society at a broader scale and are great at demonstrating this. Visit the NASA webpage and you will find a list of 20 innovations that would not exist without space travel. And we can all easily relate to these. And I love this picture called Earthrise, taken from the moon. What a contrast to our planet, which is teeming with life and where there are so many discoveries still to be made. Our ambitious mission will equally require a lot of creativity, high-tech and innovation, and a great deal of thinking outside the box. And given the great challenges that we are facing today, such as global and environmental change and rapid biodiversity loss, there has never been a more pressing time to embark on such an endeavor. And think about what we will be able to achieve. What will be the 20 things that would not exist without our work? But for now, let's look at the task. How many Australian plants are there left to be discovered and described? This slide gives an overview of the number of Australian plant species described and an estimate of what there is likely left to be described in the different taxonomic groups. As we can see, the Australian flora is already quite well described, and since this report, more progress has been made. The botanical exploration of the Australian flora is really quite a success story. After 250 years of botanical discovery, we are at 90% completion. However, this does not mean that it's going to be an easy task. The Pareto principle tells us that usually it takes 20% of effort to obtain 80% of the results, which means for our remaining 10%, there's still a lot of effort required as we are now left with all the hard cases. An additional layer of complexity is added as we are dealing with a moving target, just like the infamous Hydra was for Hercules. With new technological development and new understanding, the bar is continually raised, and our scientific endeavor requires more, not less effort. Of course, with the reward of an increasingly accurate picture of Australia's real biodiversity. A good example are genomic and epigenomic studies, which increase our understanding of phenotypic diversity. And we should also keep in mind that Hercules was able to defeat Hydra against all odds. But because there's still a lot of effort required, we have to be prepared to be asked tough questions, such as, aren't 90% good enough? However, herein lies the opportunity. The Australian taxonomic community is well placed to demonstrate the value of a nearly fully explored biota. We are in a great position to develop and deliver new, exciting, user-friendly tools and gadgets as well as services to the nation, and with our foundational work enable other exciting research, development and applications. In a recent study of taxonomic capacity in Australia, the Australian Biological Resources Study found that there is a comparatively healthy taxonomic workforce for Australian plants. The study also identified that there are gaps in certain taxonomic plant groups that need addressing, and that our workforce is aging and succession planning is required. However, with some adjustments and renewed investments, we should be in a good position to tackle the challenge. And we are also doing well at a global scale. The Kew report on the state of plants worldwide shows that Australian plant taxonomists are highly productive, with Australia ranking among the 3 source countries for plant species new to science. The Australian plant taxonomic community also has an excellent track record of early adoption and implementation of new approaches and tools. A prime example are the online taxonomic resources that our community has created in a national and collaborative effort over the past years, some of which as world's first. With the Australian Plant Name Index, we have a continually updated list of Australian plant names and publication details. We have an image database of Australian plants, and we have the Australasian Virtual Herbarium, which holds the digitized herbarium records which feed into the Atlas of Living Australia and other biodiversity portals. Images of Australian type specimens are delivered through JSTOR Global Plants, and many of our identification keys for Australian plants are available online. And of course, there is the Flora of Australia, which has moved to an online flora. These online tools greatly enhance and speed up our taxonomic work. As part of our preparations to accelerate the rate of species discovery and description, let us have a critical look at these tools and resources and see how we can increase their functionality, which data or resources can be further curated or added, and what is still missing. To achieve our goals, we will need the sharpest research tools possible. And let us also critically examine our outputs in another regard. In which form do we need to deliver our outputs to maximize the chance to create the impact required to secure the support and resources for our mission? I would argue that many of our online tools and resources are mostly catered to the botanically initiated and hold many stumbling blocks or barriers for users with less specialized background. This slide shows us that in order to achieve impact, our work needs to be taken up and adopted by others. We have a high control of our products, of course. However, we do not have control of the impact they have on economy, environment, and society. However, this path is bridged by the uptake, adoption, and consumption of our work. This is something that we can still influence, tailoring our products to the needs of our stakeholders as best as we can. Many of our stakeholders want to have answers to quite fundamental questions about biodiversity. What species is this? Where does it occur? Is it common or threatened? Is it edible or poisonous? Is it a native or a weed? And they want to have a quick and reasonably reliable answer without having to fight their way through jargon and other complexities. In order to be successful, we will need to add this little bit of extra effort to deliver products and tools that can be used more broadly. Think of the phone ID apps that use image recognition to return a result to the user without the necessity of the user to know what knowledge and technology drives this app. Let us widen the circle of stakeholders which we consider for the uptake of our products, acknowledging that most people will not be in the position or willing to undergo botanical study in order to obtain an answer to their questions. So let us have a good look at who our stakeholders are outside our immediate stakeholder group and put in effort to deliver products that cater well for their needs and level of skill and knowledge. We will need to devote more time and energy to the outward-oriented component of our work, because if we do this well, we will achieve the impact needed to enable us to do our work with the support and resources required. What would really help our cause is a service-oriented center, a one-stop shop for biodiversity diagnostics, which caters to the different needs of our stakeholders, from research to government agencies to our broader user community of biodiversity knowledge. Services would include an identification portal which can use blood images and other user-provided information, such as morphological traits and localities, and which guides the user through a clever pipeline to arrive at the identification. Taxonomy Australia has already developed a roadmap on how such a portal could be built. Further, it could include a rapid molecular taxon identification service based on querying molecular databases built at large scale from our collections, and not necessarily requiring the user to have any background in genomics, data analysis or interpretation, and in that way perhaps could be similar to genome services for human genomics. Services could extend to rapid biodiversity assessments and environmental monitoring, for example, for government, mining companies or developers. And of course, it ideally would also be a go-to place for taxonomic training, be it face-to-face or via online tutorials and webinars, and catered to different skill levels. In order to develop and deliver the required services and tools, a dedicated team of taxonomists, molecular biologists, modelers, bioinformaticians, programmers and developers is required at the back end of such a center. What form the center should have is to be discussed and developed. However, it is clear that it can only be successful if it has large multi-institutional buy-in and can count on the collaborative effort of the taxonomic community. Now, what is required to facilitate a step change in the discovery and description of the remaining Australian plants? We all know that business as usual is not going to take us there. So here are some points for discussion. First, the large-scale digitization of taxonomic resources. As pointed out earlier, the resources and tools built by the plant taxonomic community are at the heart of what we do, and increased accessibility through large-scale digitization will greatly enhance the utility of these tools. For example, the imaging of herbarium specimens beyond the type specimens, and also the imaging of specimens of related floras. Better workflows and feedback loops for data curation for increased accuracy, and here specimen IDs and distribution records are a very good example for this. But also greater discoverability of these resources is needed, for example, through common search engines. And I think a weak point is still the accessibility of taxonomic publications, which can considerably slow down progress and hamper the uptake of taxonomic work. How can we increase access to these primary outputs of our taxonomic work? Here we need to creatively think about ways how this could be achieved. Another important building block to achieve a step change in discovery and description is the generation of genomic reference data of Australian plants for resolving higher level relationships and placing specimens into a phylogenetic framework, down to population level datasets to resolve species delimitation or assess genomic diversity in threatened species. We are just entering an exciting new era in which the generation of such genomic datasets is feasible at large scale, based on our valuable collections material, with marker sets that have increased resolution and which are suited to build compatible datasets at broader scale as well as lower taxonomic level. Increasingly efficient and economic approaches and pipelines are being developed, such as high-throughput collection genomics pipelines by CSIRO's Environomics Future Science platform. And national and global initiatives are underway to build such genomic reference datasets from collections material, such as the Genomics for Australian Plants initiative, which will generate data for each Australian angiosperm genus as its first short-term goal. Also, with our data being increasingly digitally available and shareable, we can make more use of automated analysis pipelines and workflows, greatly enhancing the efficiency of our work. A great example is genetic and genomics data that can be analyzed through clever and customizable analysis pipelines, greatly speeding up the process. More development of such tools, as well as building the necessary skills within our research community, are required to take advantage of programmatic data handling and analysis. Further, it is time that we fully embrace and take advantage of the possibilities of artificial intelligence and machine learning to assist us with the huge task at hand. These approaches will rapidly develop further and offer exciting new opportunities for our community, for example, for the rapid capture of traits from specimens or for the development of image-based identification tools. And as we are a nationally and internationally dispersed research community, the use of online collaborative spaces and early data sharing will increase in importance and uptake and again facilitate a step change in our work. Lastly, digital learning opportunities and training tools for our community and our stakeholders need to be offered at a larger scale to increase capabilities nationwide more timely and rapidly, and ensure that skills are passed on between generations, and that for both ways. And with these ideas and discussion points, I would like us to enter the conversation at both ends. The inward focused end to look at what our needs are to accelerate the rate of species discovery and description, as well as the outward oriented end to assess what our stakeholders need from us to use our outputs most efficiently. And only by carefully considering both perspectives, we can be successful in our ambitious mission and deliver the greatest impact to benefit society, environment, and economy. With this, I would like to thank you for joining me today, and I look forward to our discussions. Goodbye, and thanks for listening.
So thanks, Katharina. That was a great talk. I really liked that, and I particularly liked the image you put up of the blue Earth from the moon and the point that there is just so much biodiversity and it's so important to understand it. That was a really nice touch. So I've just got a couple of questions that came to my mind when I was listening to that, and I'm hoping that other people listening to this will ask you questions through the comment box at the bottom of the presentation. So a couple of things. So this idea of an automated pipeline. So we've got all this informatics, we've got all of this vast amount of data coming from genomics, and we desperately need to automate a pipeline because the work involved is huge. But it seems to me, and correct me if I'm wrong, that every time we automate a pipeline, the methods and the technologies change, and we need to build a new pipeline. Is there a way out of that?
I don't think there's a way out of the need to update pipelines, but there are now really nice, depositories for scripts that are annotated, and you can always use those and build a new pipeline out of these or a new branch, and it's all very well documented. So I think this will be an important part of it, that we all use those script depositories where the scripts are carefully annotated and therefore usable and changeable and updatable, and like building blocks that can be plugged in or taken out of it as well. Yeah.
So the important thing is that within Australia, say if someone annotates and updates a script, then everybody else should have access to that.
Yeah,
rather than people reinventing the wheel all the time.
And exactly, and instead of just having it on your own computer and maybe sharing it among your colleagues, it would, it's really important that we use those, public depositories where there's a version controller. This is a very important part of it, and that everybody can see what has been done, and it's nicely annotated and what the changes are. And you can at any time go back to a previous stage if you want to take it from there.
Mm.
You, for example, work on an older computer or an older pipeline.
Do you have any idea how many of these scripts are on personal computers and how many of them are in public repositories?
No, I haven't.
Yeah, sure. But clearly we need to deal with that. I agree.
Yeah, and another important point is, of course, that we all upskill our abilities in reading scripts and doing a bit of programming, because even the best ready-made script, if you can't read it, then you get stuck very quickly. Yeah.
Another part of the problem, of course, is that we sort of almost all have to be experts at a whole range of things, from taxonomy to genomics to computer programming or scripting, in order to do this. And so being able to really separate those roles and having an informatics support layer that can deal with those sides of things so that the taxonomists can be left doing taxonomy, that seems to me to be important.
Yeah, I think so as well. We would all spread ourselves too thin if we try to do everything, but it's about the team that that works together and has the different skills.
Yep. Another question. So you mentioned the large-scale digitization of taxonomy resources. So anyone from, say, the invertebrate or the fungi world listening to your presentation will probably say, well, those botanists are pretty bloody well off. They've got all of these resources already digitized. What do you reckon are the next big steps? We've got almost all of our Australian specimens are digitized in the Australasian Virtual Herbarium. A lot of the literature is in VHR, a lot of the types in many overseas and all Australian collections are in global plants. That's fantastic, and entomologists would kill for that. What are the next things that we should do in this space, do you reckon?
Well, as I mentioned, I think that imaging our herbarium collections more fully would be really important. For example, if you work with distribution data or with the herbarium records and you find some outlier, a quick look at the herbarium specimen will tell you immediately, oh, this is a wrong ID, or it's a wrong transcription, and we could fix those errors much quicker if we had that all available. And similarly, if you find something you think is new and you had all those records available at your fingertips, you could really see, is this something that lies, is within the variation of this taxon, or is this, something new? It's much, I think it would speed up our work. But the other thing that I think would be really exciting would be more three-dimensional imaging. And that idea is from, image recognition of human faces, which also required, model of the head. And I think if we had models of flowers, typical flower shapes, three-dimensional, and had more three-dimensional pictures, that whole space of automatic image recognition would also be much more workable. And one prime example for that would be, of course, fresh flowers, but also our spirit collections, which often have type specimens that are only available as a flower, and to have those three-dimensional and then be able to do not only taxonomy, but also other exciting studies with those.
Yeah, because of course the other side of it is, there's a double-edged sword. The fact, for instance, that all of our specimen records are digitized, in the sense, that the record is digitized. They're not image, but they're digitized. Actually causes a problem. So every time that I do a revision of a species group in Herbertia that I work on, I know that I can curate the Western Australian herbarium specimens correctly, but there are specimens of Western Australian Herbertias in most other herbaria. And of course, every time I do a revision, many of those specimens immediately become wrong. And so their record is immediately wrong. And at the moment, the only way of dealing with that is for me to physically travel to every other herbarium and curate their collection. Yeah. Somebody there to do it, but they've all got too much to do anyway. Yeah. So being able to have a completely online visual herbarium as part of the virtual herbarium means that I could sit and do that. And you're right, that'd be great.
Yeah, and also, the person who does the revision is in the best position to really quickly go through these and sort them into the right boxes. Whereas if the curators try to apply the new taxonomy, it takes them a long time. So I think by having those available digitally would not only enhance the work that we do, but also then the records that we have available online that are automatically up to date and usable. Yeah.
Yes, because we're sort of in a bit of a halfway point at the moment. We have digital records, but like I said, many of those become incorrect records when some taxonomy changes. And so we're not yet at the point where we can make sure that all of our records remain perfectly current in the face of changing.
Without being able to see them, you wouldn't know. You might know maybe from a distribution that now wouldn't fall within that taxon anymore, but you still need to have a look at
it. Yeah, sure. So last question. So you mentioned online collaborative spaces, being able to really increase our online collaboration. And I think this coronavirus, of course, has made us all think much more about online collaboration. So can you say what would be one thing that we could do that would improve your work in setting up online collaboration spaces?
Well, of course, in the molecular space, it is still a huge problem to work together on large data sets. The data is very, heavy in the sense that you can't send it around by email. And so to have access to data sets that are analyzed in different ways, and people can take portions of the data and pre-analyze that with their data set, is still not so easy, especially with the boundaries between institutions and the permissions to gain access to servers and something that we are wanting to work out with the Genomics of Australian Plants project. So if we would be able to set that up for that project, then that could be helping other projects as well.
So something like the Cypress Gateway, but more collaborative still.
Yeah, because it would have to have, again, something like a version control. So the different data sets, for example, in target sequence capture now, you can run them through different pipelines and get different results, and they suit your different questions,
right.
at which level they are. So you would need to have the documented, how did you arrive at this data set, and also share the source data. And yeah, so it needs a lot of documentation around the data sets so that they are more shareable. Yeah.
Right. Yes. Yep. Good point.
Yeah.
Katharina, thanks again for preparing that talk and giving such a good talk. And like I said, I'm hoping that other people listening to this will use the comments box below so that we can have a more live question and answer session between you and people with other questions.
Yeah. Okay.
So good on you. Thanks very much, Katharina.
Thank you. Bye.